Thank you, Jeff. Dr. Gross is one of the leaders in the department. He's leading the research efforts in the department, and very important to what's happening currently at the Department of Ophthalmology at the School of Medicine. So I'm going to give you a question here. I've got to ask, why the zebrafish? Why the zebrafish to study diseases of the eye?
It's a great question. We've worked with zebrafish for a while now. So I started using them when I was a postdoc with John Dowling at Harvard. So John kind of had a long-standing history of using lots of different animal models to study the retina and study the eye. And his lab moved into zebrafish many years before I joined the lab. And there are lots of people around the world who study the eye and fish and other organs and other tissues as well. So there are several reasons for this. The eye in particular, it's a great model for studying eye development and eye diseases. So the zebrafish eye is really similar in structure and in function to the human eye. So the cells we're studying in a fish have immediate relevance to those in humans as well, right? If we want to study a disease, we need to have the same cell types and we need to be studying the same processes for it to be relevant. So for our work, that's, I think, number one, or the first importance is that it's really human relevant and the biology is relevant to humans. But there are also a number of other attributes that are pretty important. So they develop rapidly. They have lots and lots of babies. So, numbers are important when you're doing science and getting the most bang for your buck. So we're able to study lots of animals at the same time. They're transparent as embryos. So it's really good for microscopy and for imaging. So part of the lab is interested in developmental biology, just how the eye forms early in development. And you can see it happen in real time. And you can image cells moving, you can image the whole eye moving, you can image the structures, so we can make transgenic animals where we express, for example, a fluorescent protein in a particular cell type, and we can watch that cell type form in real time. So the imaging capability is huge in zebrafish. You can do genetics, so you can look for mutations, you can make knockouts, you can eliminate the function of a protein to model a human disease and understand the biology. And also, I think what is really exciting, certainly in our lab over the last 5 or 6 years, is studying regeneration. So zebrafish have a remarkable ability to regenerate tissues after they're injured or damaged. And this is certainly in the eye, but in the heart, in the kidneys, in a lot of different organs. So for the eye, we can start to understand how retinal injuries are resolved. So if you have an injury in the retina, the fish are able to regenerate the neurons that are lost. Similarly, there are pigmented cells at the back of the eye that help support the retina, called the retinal pigmented epithelium. Those are actually the cells that die first in age-related macular degeneration. So we're really interested in studying regeneration of those cells as well. And the fish, if you eliminate those cells or kill those cells, they come back really quickly. So it's a great model for understanding intrinsic regeneration. So the ability of the animal to sort of fix its own damage, and then understand maybe why humans can't do this. And are we, in the future, might we be able to stimulate some processes to recover lost neurons or to recover these pigmented cells or really any other organs and tissues in the body. So they're a great model for a lot of things. It's a long answer, but you can study development, you can do genetics, you can do disease modeling, you can study regeneration, lots of good biology.
And if I'm not mistaken, I think they're cheap, right?
It's for working with animals, we can house tens of thousands of fish pretty easily. And Pitt has really one of the largest zebrafish facilities in the world. So that's also a kind of an exciting thing, that the scale we can do here is quite grand.
I know you don't catch your own, but they
never had a fish in my life, actually. Never tried to keep them alive outside the lab.
Very, very neat. It is a very interesting model to study. So, you've been doing this a while now. I mean, I think he is still young, Jeff, but But you've been doing this enough that you're a senior member of the team for sure in terms of research experience. So to date, what would you say are your greatest contributions to science? What really would you say, okay, this is going to do something for the world here?
So it's a great question and a bit of a humbling question, I guess, to think of your career in that way. We're actually about at the 15-year anniversary of having a lab in another week or so. I started my lab in Texas on August 15th of 2005. So we're hitting 15 years. Greatest contribution. I actually say this might not be the answer you're expecting, but I would say the people are the greatest contribution from the lab. So as you said in the introduction, there have been, at the undergraduate level, we've had probably 50 undergraduates now who have passed through the lab, 8 PhD students that have graduated with several more in the lab, many postdocs, and they're all involved in science. And I think for me, that's what I'm most proud of, actually, that's come out of the lab. I'm proud of the work that they've all done. These are really wonderful, creative people that have come to the lab. And I run the lab in a really hands-off way where I want them to come up. They're the drivers, they're the intellectual forces in the lab, and they come up with ideas. And we're very blunt in the lab. We tell each other when it's a good idea and when it's not a good idea, and really hash these things out. And the people that have come out of the lab are doing really amazing things. Some of them still work in the eye, some of them don't, but all of them are doing great science. They're all, many of them are involved in teaching. So they're at other universities, they're graduate students, they're medical students. So they're kind of continuing science education in the world, which I think is extremely important, that people continue to do this. So yeah, I'm proud of the science we've done, and I'm happy to talk about that too. But I would say, I think the biggest contribution is the people that have come out of the lab and the science they're doing. And it's really amazing work.
Well, that'll certainly be a long-lasting legacy, no doubt about it. And that is core to what we do. And and you are humble because I know there's been a lot of incredible discoveries that your lab has made, and in terms of regeneration and things that you found, and new pathways and new directions, and so maybe share one of those with us.
Yeah, no, I'm always happy to talk about our science too. A great example, I had a former PhD student in Nick Hanovice in the lab who started what now I think is probably the focus of almost half of our lab. And that's, again, studying the regeneration of these pigmented cells at the back of the eye, the retinal pigmented epithelial cells. So Nick, as a PhD student when he was in the lab, kind of proposed this idea to look at whether zebrafish were able to regenerate these cells. And it actually wasn't known in really any animal whether you could stimulate these cells to regenerate. So he devised kind of a clever way to ablate or kill about 2/3 of these pigmented cells at the back of the eye, and then look at whether they regenerate. And as biology would have it, or as evolution would have it, the zebrafish is indeed able to regenerate these. So now we've been studying that process and trying to understand how they first respond to the injury. So what tells them something's wrong? What are sort of those injury responses? And then how do they resolve that injury? And then how do they regenerate the lost tissue? And there's some extremely exciting work to me that's kind of emerging from the lab right now, looking at the immune system. So I've got a terrific scientist in the lab, Dr. Lindsay Leach, who's kind of been doing this work and looking at how components of what are called the innate immune system. So cells come in, immune cells come in and start to clear some of that debris. And through various signaling and cell-cell interactions, they actually stimulate this tissue to regenerate. We're trying to understand how that happens, because again, in the human eye, there's an immune component to diseases like age-related macular degeneration. The immune system can be sort of helpful in some ways, and it can be harmful in some ways. So we're starting to think about ways we might be able to push that response into the helpful direction in the human eye, maybe to stimulate regeneration, but even sort of at a more immediate goal, just to limit some of the damage that occurs. And we're learning a lot about that biology in the zebrafish, and that comes from this project that came from, you know, a couple of really creative people that had great ideas in the lab and pushed forward to get those projects going and really develop them into something that I think we can run with now for many years.
Well, that's actually a great story. Um, so, you know, I, this is of interest to me because I'm you know, a fan of our local community in the city that we live in. But you came to Pittsburgh in 2015, become the director of the Fox Center for Vision Restoration. What convinced you that Pittsburgh was the right place for you and your family?
Yeah, so first off, Pittsburgh's a great city. So the city did a lot of the selling non-scientifically. I mean, people are friendly here. It's livable. There's lots to do. I'm a huge, huge, huge professional baseball fan. So even though the Pirates aren't very good, I get to go to baseball games all the time, which is a big part of, you know, the summers and fall for me and my family. You know, the science at Pitt is phenomenal, and coming to the ophthalmology department was exciting to be surrounded by other people who are really thinking about the eye, how it forms, how it functions, diseases, having clinicians who are engaged in research, and then really this amazing cadre of researchers that we have who are asking and answering all these exciting questions. So that was, you know, on its own was the opportunity to sort of push our science in new directions was exciting. Taking over the Fox Center was really exciting. So we have a terrific donor, Mr. Lewis Fox and Dorothy Fox, his wife, who started this center. And you know, we've really kind of focused and honed that into looking at optic nerve regeneration, which is a tremendously exciting topic for people who have suffered injuries and lost the optic nerve or lost the function of the optic nerve, or in glaucoma where ganglion cells that generate the optic nerve start to die. So taking the Fox Center and really pushing the science and the teams forward was an exciting opportunity for me. I really like working with people, and we've got great scientists in the Fox Center. I would say that what really sealed the deal was a conversation with our former dean, Art Levine, who is a very, very compelling salesman at the science and the excitement at the University of Pittsburgh and the medical school. So when I visited and had a conversation with Art about the future here, it's, you know, it just seemed like the right place for the next phase of our work, and that kind of caused us to pull the trigger and move here.
Yeah, we're always. I've been very happy that you have. It's been, you know, a good, you know, experience for me as well working with you. So you were recruited to advance research in regenerative medicine, which of course is the main focus of your work. But you have taken on a very large role as the, you know, the vice chair of the director of research in helping Dr. Sal build one of the largest teams of science and vision research, quickly growing to be probably one of the largest in the country, and soon to be, you know, maybe even bigger than that. Did you ever imagine you'd be doing something like
this? No. No, I didn't. I mean, when I was in Texas, I had sort of started to take on some more administrative roles and you know, running programs, and I really enjoy that. I mean, I think when you have great people, you know, they're going to do great things if the structure allows them to do so. And the faculty we have are amazing. The faculty we've recruited are amazing. So working with them, it's really, you know, it's been a treat. I get to learn about their science and talk to them. You know, you all at the foundation do wonderful work. So it's great kind of serving as a bit of an interface between some of the scientists and you all, and helping foster this environment that we're growing. Dr. Sahel's vision for the department, for the new Vision Institute and hospital that we're building, and really the, you know, the city of Pittsburgh and region are, it's an amazing and extremely ambitious vision. And I think, you know, again, having that ambitious of a goal and being part of something way bigger than my lab and our own research is, you know, it's really exciting. And maybe I'm coming back to an earlier answer of being proud of the people in my lab, but I think also, it really is the scientists. I mean, the people that we have, the clinicians, the researchers, their teams, everybody's completely vested in what we're doing here. And I don't think I would be able to serve in this job as the research director if it weren't for those people. They make the job really easy. People who want to be here, they're striving to do the best science they possibly can. And all I have to do is kind of help them navigate a little bit through the intricacies of the university and some of the things that come with that. So it's a fun job because they're doing amazing work, and they're going to continue to do amazing work. And I think Dr. Sahel has a real knack for finding people who, work well together in sort of a big collaborative environment. They have their own science, which is excellent, but they're really willing to pitch into some of these bigger projects that, that people couldn't, you couldn't really do some of these things in your own lab, but if you've got 3 or 4 people around you who are excited about it and are creative and good scientists, it enables these sorts of things to happen. So no, I didn't envision I'd be doing this, but I'm really glad, and I feel lucky to work with him, lucky to work with you all, lucky to work with our donors and our constituents in the city who are engaged in what we're doing. And then the scientists here, everybody's completely aligned towards this goal, and I think we can get there.
Fantastic. Well, thank you. It's been great to give everybody a chance to get to know what I do know, which is, what a terrific leader you are, what a great, passionate scientist you are, and again, what an asset you are to the department and to Pittsburgh. So, thank you. And we'll say goodbye and have a great day.
Thank you.